Good day. Okay, today let's take a look at your extracellular matrix. This is a part of your activity in your laboratory for histology. Okay, and so let's take a look at this. Of course, under this presentation, we'll take a look at your extracellular matrix together with your collagen synthesis. Of course, collagen is part of your extracellular matrix together with your glycosaminoglycans and then collagen and your elastic fibers. Okay, and so with this, let's take a look first at your basement membrane. Okay, and so please note that your basement membrane, this is a thin, specialized sheet of extracellular matrix. Okay, and so please note that this is the interface between your connective tissue or the underlying tissue and your squamous epithelium. Okay, and so with that, it is a thin matrix, a thin specialized sheet of extracellular matrix. Okay, and so with this, in your basement membrane, there is no cells present. Okay, and so this is located at the interface between epithelial tissues and your underlying connective tissue. Okay, and so with that, it will separate the cellular parts of your tissue. Please note that your epithelial tissue, this is made of individual epithelial cells, while in your connective tissue, there are also cells present. Okay, and so you have your fibroblast, your chondroblast, and the rest of your fixed cells present in your connective tissue. Again, in your basement membrane, there is no cells present. It is considered as a cellular part of the tissue. Okay, and so this is essential for tissue structure and function accordingly. Yes, that is for your basement membrane. Okay, and so your basement membrane, you have types. You have your basal lamina and your reticular lamina. Your basal lamina, this one, you have your lamina lucida. This is a thin, clear layer adjacent to the epithelial cells, while your lamina densa, this is a dense layer that is rich in type 4 collagen and laminin. While for your reticular lamina, it lies beneath your basal lamina. Okay, and so for your basement membrane, again, you have two layers. You have the basal lamina and your reticular lamina. Okay, your reticular lamina lies beneath the basal lamina, and this one contains types 3 collagen, and this one will anchor the basement membrane to the underlying connective tissue. That is for your basal lamina, and your reticular lamina has the two layers of your basement membrane. Okay. And yes, let's take a look at your extracellular matrix. This is an organized meshwork of macromolecules that surrounds your cells. Of course, you have your macromolecules present, you have your glycosaminoglycans, your ground substance, your glycosaminoglycans together with your proteoglycans and your multi-adhesive substances. Okay, and so of course in your extracellular matrix, it consists of protein fibers and ground substance. These protein fibers will include, of course, your collagen, your reticular tissues, together with your reticular fibers, sorry, together with your elastic fibers accordingly. And of course, for your ground substance, it is a complex of anionic hydrophilic proteoglycans, glycosaminoglycans, and multi-adhesive glycoproteins. Okay, of course, your what you call this extracellular matrix, this is where your animal tissue cells are embedded. Okay, and so seen in your extracellular matrix will be your cells, like your fibroblast and the rest of your fixed cells in your connective tissue. Okay, and so this is an illustration, a diagrammatic illustration of your extracellular matrix. Okay, and so of course you have here for your ground substance that is made of your ground substance, you have here the presence of your glycosaminoglycans, proteoglycans, and your multi-adhesive glycoproteins. And then you have your protein fibers, you have your elastic fibers, your collagen fibers, and your reticular fibers accordingly. Okay. And again, another illustration of your extracellular matrix. Again, you have here your fibers, you have your collagen, your elastic fibers, and then your ground substance, you have your glycosaminoglycans and your proteoglycans. Okay, and so accordingly, what is the function of your extracellular matrix? Okay, and so with this, for your extracellular matrix, it provides a scaffold for tissue organization accordingly. Yes, it will provide a scaffold. Also, it will provide support. Okay, and so with this also, it determines the mechanical properties of your tissues. And yes, it also functions for movement. With that, it facilitates cell attachment and movement accordingly. And yes, also it guides migration of cells during development and wound healing. And it will also influence cell differentiation, proliferation, and survival accordingly. And yes, regulation. Extracellular molecules accordingly can bind to growth of factors. Okay, and so controlling their availability accordingly. Okay, and so that is the function of your extracellular matrix. Okay, your components. Okay, and so as what I said earlier, you have your protein fibers, you have the collagen, your elastic fibers, and then you have your reticular fibers. Okay, and so for this part, we'll take a look at your collagen fibers. Of course, with this, it provides tensile strength. Okay, and so collagen fibers is abundant in your connective tissue, especially in your dense connective tissue. Your elastin, okay, and so that is present in your elastic fibers. This one has a rubber-like properties that allow tissues to be stretched or distended, and yes, they return to their original shape. Okay, and so of course your skin, you have accordingly you have your elastin present in that, so that if you press it and then you remove the finger, when you press the one, it will return to its original structure. Okay, and so that is attributed to the presence of your elastic fibers in your tissues. Okay, and so and then aside from that, again, you have your ground substance. Your ground substance, you have here the presence of your glycosaminoglycans, proteoglycans, and multi-adhesive glycoproteins. Okay, and so for your glycosaminoglycans, they are long polymers of repeating disaccharide units. Okay, and so usually these disaccharide units are hexosamine and uronic acid. Okay, and so usually these disaccharide units present in your glycosaminoglycans, they are usually hexosamine and uronic acid. Okay, and so for your proteoglycans, this one consists of a core protein. Of course, proteoglycans, that is a mixture of carbohydrates and protein. Okay, and so it consists of a core protein to which are covalently attached various numbers and combinations of sulfated glycosaminoglycans. And then you have your multi-adhesive glycoproteins. This one possess multiple binding sites for cell surface integrins and for other matrix macromolecules. Again, for your ground substance, you have your composition, you have your glycosaminoglycans, proteoglycans, and multi-adhesive glycoproteins. Okay, so let's take a look at this components of your of your ground substance one by one. Your glycosaminoglycans, they have again, okay, and so the hexosamine component, you have your glucosamine or galactosamine. Okay, and so for your hexosamine, this one is your repeating units of your disaccharide, as what we mentioned earlier. Okay, and so and this hexosamine component is either glucosamine or galactosamine. Okay, and for the uronic acid, that is either glucuronate or iduronate. Okay, and so that is for your uronic acid. Okay, and so please note that your please note that your glycosaminoglycans, they have a 10 to 40 kilodaltons molecular weight. They are sulfated and they possess negative charge. Okay, and so please note that this high negative charge of your glycosaminoglycans forces your glycosaminoglycans to an extended conformation, and with that will cause them to sequester cations as well as water. Okay, of course, this feature, they can sequester cations as well as water. This feature will provide glycosaminoglycans with space filling, cushioning, and lubricating functions accordingly. Okay, and please note that glycosaminoglycans, these ones are synthesized in your Golgi complex. Okay, and so examples of glycosaminoglycans, you have dermatan sulfate, chondroitin sulfates, keratan sulfate, and your heparan sulfates. These are actually your major glycosaminoglycans. But then, of course, let's take a look at your glycosaminoglycans one by one. Okay, and so please note that your hyaluronic acid, or this one is otherwise known as your hyaluronan, is the largest among your glycosaminoglycans. Okay, it is located in your umbilical cord, synovial fluid, vitreous humor of your eyes, and in the cartilage. Okay, and so this one accordingly, unique among your glycosaminoglycans, your hyaluronan or hyaluronic acid is synthesized directly into the extracellular matrix by an enzyme complex known as hyaluronan synthase. Of course, this hyaluronan is located in the cell membrane of many cells. Okay, and so that is why it is present in the cell membrane. It can directly synthesize hyaluronan in the extracellular matrix. It does not need to be it does not need to be secreted via your vesicles. Okay, and so because of course the enzyme needed for the synthesis of your hyaluronan is present in the cell membrane. Okay, and so it is not present in the cytoplasm or not present in the cytosol. Okay, another glycosaminoglycans, you have your chondroitin sulfate. This one is located in your cartilage, bone, cornea, skin, notochord, and aorta. Okay. Other Another one, you have your dermatan sulfate located in your tendon, skin, aorta, in the adventitial layer of your aorta. And then you have your heparan sulfate located in aorta, lungs, in the liver, and in your basal lamina. And your keratan sulfate, it is located in your cartilage, nucleus, nucleus pulposus, annulus, and fibrosus accordingly. Okay, and so these are your glycosaminoglycans and their locations. Moving on, let's take a look at your proteoglycans. Okay, and so this one, they have attached glycosaminoglycans that comprise a greater mass than the polypeptide core. Okay, and so the core here is a protein. Your proteoglycans accordingly become bound to the hyaluronan by link proteins. Okay, and so you have here your most commonly seen, you have your perlecan. This is a proteoglycan. This is the key proteoglycan in all basal lamina. And then you have your agrican. It has a core protein heavily bound with chondroitin and keratan sulfate chains. Okay, please note that your agrican, again, this one is your agrican, this one is abundant in the cartilage. Okay, and so agrican in the cartilage is uh complex or attached with hyaluronan to form your agrican hyaluronan complexes. Okay, and so of course this complex will fill the spaces between collagen fibers and cells, and with that it contribute greatly to the physical properties of your tissue. Of course, that is your cartilage. Another proteoglycan is your decorin. You have a few glycosaminoglycan side chains that bind the surface of type 1 collagen fibrils. And then another proteoglycan, you have your syndican. This one has an integral membrane core protein providing an additional attachment of extracellular matrix to the cell membranes. Okay, and so that is for your proteoglycans. Moving on to your multi-adhesive glycoprotein. Okay, they are large molecules with branch or oligosaccharide chains that allow adhesion of cells to their substrate. Okay, and so you have your laminin. This is a multi-adhesive glycoprotein, which that has binding sites for integrins, type for collagen, and specific proteoglycans accordingly. Your laminin, this one will provide adhesion for epithelial cells and other cells accordingly. Please note these ones are multi-adhesive glycoprotein. Okay, and so it will again help to seal or to connect with each cells. And then you have your fibronectin. It has binding sites for collagen and certain glycosaminoglycans, and with this forms insoluble fibrillar networks throughout your connective tissue. Okay, and so please note that your fibronectin substrate provides specific binding sites for integrins, and is important for both for cell adhesion and cellular migration through the extracellular matrix. Okay, that is for your multi-adhesive glycoprotein. Okay, and so let's move on to your collagen synthesis. Okay, and so your collagen, this one is a core part of your connective tissue. But then let's take a look at how this one is being synthesized. Okay, and so of course, your collagen is synthesized primarily by your fibroblasts. Okay, and so your fibroblasts, these are resident cells of your connective tissues. Okay, and so of course your what you call this, your rough endoplasmic reticulum, the organelle rough endoplasmic reticulum synthesizes pro-collagen alpha chains. Now it's not actually the rough endoplasmic reticulum, but the ribosomes present, the ribosomes attached in your rough endoplasmic reticulum, these ones are responsible in synthesizing pro-collagen alpha chains. Okay. Okay, and so with that, in your rough ER, there is a production of your triple helix that is produced by aligning and stabilizing three pro-collagen alpha chains. Of course, these ones are stabilized by the sulfide bonds at their carboxy terminal. Okay, and so after that, of course, the production of your triple helix, it is released, then cleaved to Iran-like pro-collagen molecules. Please note that different combinations of pro-collagen alpha chains produce the various types of collagen with different structures and functional properties. Okay, and so for the step one, of course, there should be a gene transcription. Okay, and so gene transcription happens inside, of course, in the nucleus. You have your collagen genes. You have these ones are needed for transcription of collagen, collagen messenger RNA in the nucleus. Okay, and then you have your translation. Of course, you have here your messenger RNA translation, and this one occurs in the rough endoplasmic reticulum in the ribosomes attached in your rough endoplasmic reticulum. Okay, and in this translation, you have here your pro-collagen formation. That is the initial polypeptide chain synthesized. Okay, and so for your transcription process here, we will be taking a look at this process in your biochemistry. Okay, and so step one of your collagen synthesis, you have your pro-collagen alpha chains are produced in the rough endoplasmic reticulum and translocated in your cisternae. Okay, and so produced in the ribosomes, in your ribosomes attached in your RER and translocated in the cisternae accordingly. Okay, and so with this, it will typically have long central domains rich in proline and lysine. Okay, and so after that, in your cisternae, in your endoplasmic reticulum cisternae, you have here the presence of your hydroxylase that will add hydroxyl groups to some prolines and lysines in your pro-collagen alpha chains. Okay, and so please don't forget of the action of your hydroxylase enzyme. Okay, and so for that. And then you have here your glycosylation of some hydroxylysine residues also occurs. Please note that glycosylation here is the addition of your sugar molecules to your hydroxylated lysine residues. Okay, and so by the way, hydroxylation here is the addition of your OH group or hydroxyl group to your uh, lysine and proline amino acids. Okay, and so there is again a glycosylation of some hydroxylysine residues in various degrees of collagen types. Okay, and then you have your assembly of procollagen molecules in triple helix. You have your carboxy terminal regions of your three selected alpha chains are stabilized by cysteine disulfide bonds. Of course, with this will produce your triple helix. Okay, and so once this selected alpha chains are stabilized with by cysteine disulfide bonds. These ones will align to three polypeptides and facilitates their central domains, folding as a triple helix. Okay, with that, there is a production already of your triple helix. Okay. After that, of course, okay, with of course its globular terminal sequences intact, your trimetric procollagen molecule is transported through the Golgi apparatus and then packaged in vesicles and then secreted. Okay, and so you have here your secreted, of course, outside of the cell. Then you have your procollagen peptidases will remove the terminal globular peptides in procollagen molecules. Of course, this step will happen now outside of the cell. Okay, and so it is now outside of the cell. Again, your procollagen peptidases will remove the terminal globular peptides in procollagen molecules, converting your procollagen molecules to a mature collagen molecule. Okay, and with this collagen molecules will aggregate to form collagen fibrils. Again, these mature collagen molecules that are produced from your removal of the terminal globular peptides will aggregate to form collagen fibrils. And then after that, you have here your attachment or association of your certain proteoglycans and other collagens with the new collagen fibrils. Of course, they will stabilize also the assemblies and promote the formation of larger fibers from the fibrils. Okay, and then finally you have here the cross-linking that is catalyzed by your lysyl oxidases. Your fibrillar structure is reinforced and this assembly is prevented by the formation of covalent cross-links between your collagen molecules. Again, this one is catalyzed by your enzyme lysyl oxidase. Again, your cross-linking, your lysyl oxidase will form covalent cross-links stabilizing your fibrils into larger fibers. And this one is catalyzed by your lysyl oxidase. Okay, and so going back to your collagen, your collagen here is important for tissue strength and integrity. Okay, and so with that, it provides tensile strength to your tissues, and this is essential for wound healing and tissue repair accordingly. Okay, and so please note that in the synthesis of your collagen, at some point in the synthesis of your collagen, there is a part that needs your vitamin C. There is a part in these steps in the synthesis of your collagen that it needs vitamin C. Please note that for collagen synthesis, for collagen to be synthesized, it needs vitamin C. That is why if there is a deficiency in vitamin C, then it will also affect your synthesis of collagen. Okay, and so it will affect the integrity of collagen that is being produced. Okay, and so your scurvy, your vitamin C deficiency will produce, a defective collagen synthesis. Okay, and then finally, you have your elastic fibers. These are thinner than type 1 collagen fibers and form your sparse networks interspersed with collagen bundle. Okay, so they are somewhat smaller in number, and yes, they are thinner than type 1 collagen fibers. Okay, and so what is the function of this one? It provides elasticity and resilience to tissues. And this is found in organs that require flexibility, like the skin, lungs, and blood vessels accordingly. Okay, and so that is for your elastic fibers. And yes, that is for your extracellular matrix activity.